I can print you one at some point. Part 1. Only 2 weeks left. It's crazy. Crazy.
One of our friends just bought Kansas City ones, and they were right over the tunnel.
Oh, so we're almost done.
Like 600, yeah. They were literally right in Marshall's mama.
Yeah, they were like, what?
Yeah, see, they were right here. They were literally right there. Really, yeah.
I was like, how did you know it was his mom? Like, how does that even come about? I was like, did he like come up and like, you know, I was like, I don't know. And she's like, yeah, it was pretty cool. She's like, oh, wait, down.
How many people need a copy of that assignment? Part 1. 3?
When do you want us to do it? Oh, yeah, I have my everything done.
Yeah?
Yeah.
Hey,
that's okay. It was that one with like the.
Yeah, let's do it before lab tomorrow. All right, so see if you remember anything from last. Jeez, a long time ago, a week ago. What's different between first and second line of defense? Wait, who was that? All right. Somebody over here. Do I?
I thought we already did this question.
Did we? I don't remember doing this one. All right, looks like you have some discussing to do. Look, we had at least one person vote for every answer. It's pretty good. That's ridiculous. All right, so maybe you need to have a little discussion there.
We got all the way through to the interferon, right?
This is after the interferon part. It's the very last slide question. What else is, oh right, true, false, Venn diagram. I need to add one word to this one.
That changes it.
All right, so what do y'all think? Everybody vote? Looks like we got 13, 37, 11. All right, so we had a big shift to F, which was B, C, and D. That's not right. The answer is actually B, which is what 30% of you put.
Okay, that's
So T cells, is that first, second, third line of defense? Which one is it?
Second. No, it's third.
That's third line of defense, is T cells. What about B cells?
That's the first. No, it's really, yeah, I feel like that's more of the first. And then that's the second.
B cells are also third line of defense.
I give up.
I know, this part's hard. Specific, is that first, second, or third?
Which one?
Specific,
Third.
it is third.
It's on another.
It's a little mean because we didn't do third yet in detail.
It says right here, it's specific or cell or antibody based.
Um,
Way in the beginning.
And then antibodies, first, second, or third? It is third. So whether or not immune cells are involved, it's the difference between first and second, which brings us to the Venn diagram that you're going to work on with your group there on the tables. Actually, not individual first, because we don't have time for that. So Venn diagram between first and second line of defense. You just got one thing. Which one has immune cells? Second, which one doesn't? So there's your first difference. And I'll put, let's see, where was that one picture that helps? If you can read that, that might help too. That slide from way back at the beginning.
Oh, yeah, I see.
All the space I gave you for this one.
Would that be like the cell, like that this is cell-based and this is through?
Yeah, like immune cells.
Oh,
I think you already wrote it. Yeah.
first.
All right, so maybe check in with your neighbors there, see if they've got something you don't.
Okay. That's right. How do you copying this from here?
Well, you should think about it a little bit. Like, look at these two things and try to find, is there anything the same, anything different? Maybe glance at this one too. If you can read that, or you read it from up there. So, are you looking for something for first? So you can put some like examples of first, like skin, right? Like basically all those up. Oh, it's not on there. It's on that other slide. Sure, those would be examples. And it's confusing because it's lysozyme. It's like one letter different than the organelle, which is annoying. Yeah. They could have given it, a different name. Yes. Anything with chemicals is first.
But it still says second line of defense has a chemical system.
Oh, I think they're talking about cytokines.
So that's not for both.
No, because, I don't know. I think chemicals first. That's what I would think.
Okay, so
And I would say cytokines are second.
So there are cytokines in the second line of defense?
That's the only place. Well, they're in third line too, but they're in second line. Not
So don't
first.
put a chemical system for line 2 in the second lines.
No. I would put that for first because it's chemical barriers like acids, lysozyme. Complement would be second.
Yeah.
All right. So some similarities. What are some similarities? Non-specific. What else? They both protect you from infection. Sure. One thing that people always get it confused, locations. Where's the first line of defense? But like where in your body? Is it only outside? What about like your stomach acid? Or remember flow of urine was also?
So both would be inside, right?
Both would be inside, but only first is outside. So the first is inside and outside, the second is inside. So however you want to write that down. I think on tests, everyone always says the first line of defense is only outside of your body, but it's not. There's lots of places inside too. And then in terms of differences, basically, second line of defense is immune cells, first isn't, and then all the examples. So first line of defense, physical barriers like your skin or mucous membranes, chemicals like lysozyme, stomach acid, sweat, things like that. And then second, cytokines, inflammation, fever, interferons. Cytokines. Did I say cytokines already? I think I did. All right, so let's try this on your own first. It's a bunch of true-false questions. See what you remember. I hope you do. I'm going to go get something from the printer. The last one's a good one. Right. Make sure you fix the false ones. Did anyone correct the false ones like it says in the instructions?
I did one of them.
Okay, all right. So as you're fixing those, go ahead and check in with your group mates there. I need to pull it up. You are right. What would make sense, do you think? Right? The viruses don't want to help our immune system. Why would they make it, right?
That's how I am.
Yeah, I'm like, I can clean up poop, I can clean up puke, I don't care, just don't spit on me.
Yeah, that's how I am.
I had a lady puking on the plane yesterday. So there's some turbulence. She had this big trash bag. She's like, "Ugh."
I would be so embarrassed if I was puking.
No lie.
Why do you know? She's like, I saw her for like 5 minutes like this. I'm like, why didn't you go to the bathroom? She was like in the back row.
Before.
She don't want to get up. I know that feeling. It's like, I'm not.
Yeah, that's true.
I've thrown up on a nurse in the ER. I
Really?
realized I was going to throw up, and then it just happened. I'm like,
You're like, I'm really sorry.
didn't we just?
Yeah.
He was like clearing up blood in the ER. It was like my first clinical day ever.
Surprised you didn't quit after
He
that.
like missed it, and I was like,
Ugh, gross. All right, so first one. It's false.
Should be non-specific.
It should be not specific. What about B? Speeds up the response. C. That one's false. So you could say in response to viral infection or by virally infected cells. Either way would be right. What about D? Did I make all these false?
No.
Oh, good. E? That one's, yeah, it's false, not specific.
I changed it and I put the third line.
Or you could do that. Just have to be different.
I went,
Very true.
That's totally fine. And what about F? True. And the last one? True. Very true. Extra true. Hopefully not too many of you put false for the last one. Be concerned about you.
I'm like, whatever.
I'd rather pop a zit than get spit on.
what?
So I guess I'd rather pop a zit than get
I
spit
would.
on.
Right?
Absolutely.
Right?
So are y'all in agreement? Everyone hates spit more than pus?
Yeah.
heard like multiple people say that.
Blood, vomit, I'm fine. As soon as I start packing a
My brother used to do that to me. He'd hold me down and.
Oh, that's terrible.
It's okay, every morning my fiance would look my face and he goes like, "Oh, my God." And I went to punch him, but he moved so damn fast.
So damn fast.
I don't know why.
That's terrible.
Hates it.
Oh, I would do.
All right, so questions on the study guide.
So lot.
So many. I don't have one, so you have to tell me what it is.
I just have a problem with like when you say the B cells, there's different types of B cells. So like,
Like plasma and memory? We're talking about T cells?
no, I'm talking the B cells. There's different.
It says identify the function of the following blood cells.
Yeah.
And then it says some B cells.
B cells make antibodies.
Okay, that's it.
What about the T cells?
T cells?
Helper cells.
T helper cells kind of activate the immune response.
So they would activate B cells.
They do. So they're kind of, the T helper cells activate the immune response. Mostly like the specific, the third line of defense.
What's unique about each B cell and T cell?
What is unique about each B cell and T cell is their antigen binding receptor
Is it 'cause, like,
and their personalities.
Where are they bind, like the B cells bind on the outside or whatever the T cells bind? Binding receptor.
Just for E.
What's E?
Function of B cells and T cells.
What do you want for E exactly?
Basically, B cells make antibodies. T cells activate the immune response or directly kill infected cells. It just says T cells in general, right, on there? Yeah. So,
Okay,
What did you
so
say for the T cells?
so T cells activate the immune response, or other T cells directly kill infected cells, like virally infected ones.
I feel like you're making that a lot more complicated than it is.
So on D though, like, because I saw this thing, I was trying to look it up. So, like T cells can only connect to virus antigen on the outside, and B cells can connect around the surface.
That sounds like the same thing to me. So you said it on the outside of the cell and the surface?
The antigens right on the surface of the invading.
Oh, of the virus.
And then T cells can only connect to virus antigen on the outside.
Yeah, I would say technically that's the antibodies that the B cells make that do that.
Okay, so either
And he
way,
put antigen binding receptor.
that's all you need to know is the antigen binding receptor.
For what's unique about each one.
Yeah.
Or for
B and
no,
they both
D.
need it.
No.
So what's unique about each of those cells is their antigen binding receptor, which you'll see kind of what that means later today.
So we don't really need to know what she just kind of said too? 'Cause
You will at some point.
Not relevant.
Um,
Eventually need to know though, so like the B cells can go on the outside.
B cells can make antibodies that directly target the virus,
Darn B
And
cells.
T cells killing me.
and T cells can't. They all T cells deal with your own cells that are infected or broken.
Okay, that made a little more sense.
Yeah, that makes more sense.
B cells make antibodies and T cells do. Okay.
Yeah, they do everything else.
Yeah. T cells affect your cells, B cells affect the dying cells.
Oh my God.
What'd you say?
Cleared it all.
T cells affect your cells.
Right.
And B cells affect the viruses.
There you go.
Which is helpful.
I put in a.
Yeah, me too. This whole chapter makes sense now.
No, it doesn't. Don't lie to us.
One little part of this book makes sense now.
No, like when the tiny little part.
All right, so let's do part 2, Quizlet.
I feel like I had to just fill out half my paper.
And that's how I feel.
It's crazy. We have today, and then it's like test.
I know, you're so mean for
You're
doing
telling
that.
us.
I know. We could just have a giant final that has chapter 18 and 22 combined. It's worth 30% of your grade. didn't want to do that.
Dude, that just dumped a huge,
Let's not do that.
yeah.
All right, let's go and pass those on up. Nope, too late. This is a bad one.
Yeah. That's upside down.
Nobody asked me about the first two questions.
I thought I knew it until you handed it.
Yeah, it's in my notes and I'm like, crap, what was that?
It happens to me on the test for me.
I think that's the first time. I think every other time I've done this, someone asked about those too. But anyways, antigen, type of cell, foreign molecule, molecule produced by B cells, a receptor on T helper cells. It is a foreign molecule. What is an antibody? We said it like 5 times. It's a molecule produced by B cells.
What letter was that?
That was C. Function of T cytotoxic. Produce antibodies, activate immune system, directly kill virally infected and cancer cells, or phagocytosis. It's the third one, directly kill the virally infected and cancer cells. What is unique about each B cell? Antigen binding receptor. General function of cytokines. Bind to pathogens, activate immune system, kill pathogens. Let's activate the immune system. Right. First one was foreign molecule. All right, so let's see how far we can get on this one. This is not chapter 15. Don't worry. All right. So the third line of defense, it's adaptive and it's specific. Specific means it recognizes certain molecules on certain viruses or bacteria. Adaptive means you can get immune to certain bacteria. Like the next time you see it, your response is stronger. So you can like kind of adapt and not get sick from the same thing over and over again. And we'll see how that works. First up is our antigens. That's question 1, some Play-Doh. Let's make this, see if we can make this snappy. So you're gonna draw a big circle on your table and put 3 different shapes on the outside made out of Play-Doh. So that's question 1. Oh, and for this one, make it all one color of Play-Doh. It'll make sense later. So 3 shapes, they're the same color. So any shapes you want on the outside, doesn't matter. Give me a shape, give me a shape. I want to see a 5-pointed star. Makes me so mad. Like try to draw stars in school, and like that last little star point's always like all ugly.
Really, really long.
Makes me so mad.
Yeah, and then make a circle.
I didn't either. So upsetting.
I always had to like do the thing and then erase the lines.
No, there must be some trick that we never learned.
My mom can do it. My mom can draw. I can paint though, but I can't draw.
All right, she has 3 shapes yet? Are you hogging all the Play-Doh?
You know what? You
I'm handing it out. We're gonna do different colors.
I know. We want our antigens one color, and then we'll make antibodies another color. Oh, put on the outside.
I know. I
Okay.
just have to have a surface.
I know.
I think the dog would do like the no armband or whatever. Where the bees are like chasing him.
Here, that kind of looks like the square.
Here's my rectangle.
So stick those on the outside of your cell, and you'll be done. Cool. So like attached to the membrane, like right there.
You did a circle too.
Sorry, they're different size circles. That works. Here, here, make it a really tiny circle, so then it's obvious it's different. Boom. There you go. All right, so you got yours. Looking good. Hey, you are making a star. Yeah. Hey, it's pretty good. I don't think I could. I'd have a hard time. All right, so those are your antigens. All right, and did it say it on there? So that circle you drew, that's like an invading virus or bacteria. So based on kind of what you did there, what do you think the answer is to those? All right, so maybe chat with your group there for a moment, and we'll see.
You gotta add an S to system.
No, it's an istom.
I was like, it doesn't need to be immune system.
All right, I'll fix it just to make you happy. All right. So it is E. It is all of this. So one of the important things I think that people always get confused on is your immune system isn't responding to like a virus or bacteria. It's responding to one of those shapes on the surface. So if they, you know, those like virus spreading into the world movies, they're always like, oh no, the virus is mutated, right? There's always that line in there somewhere. So that means that shape that they developed the vaccine or your body's fighting against changed shape. And now your body doesn't know how to fight it anymore. does activate the immune system, and obviously, like you just showed me, there's multiple antigens on the surface, different shapes. All right, so next up, you're going to make some antigen binding receptors out of different colors of Play-Doh that bind to those antigens you made.
Yeah.
So we're supposed to make like a match.
Well, what do you think?
Binds, they have to have similar shape.
Well, you know, I get with that. So I'll do the triangle.
Oh, is it like the thing we did before?
Yeah.
You mean,
Square and then circle.
what did we do that for? Enzymes? Yeah. It's the same
Something
idea.
like the same type of thing? I just remember we made it a lot more complicated than it was supposed to be.
This one will be a challenge.
I know.
There you go.
Which one are you doing?
Oh, you made a little Tootsie Roll. Mm.
That one's
Yeah, you're
the
good.
other way. Okay.
Because that's on the surface, and this is like binding to the outside of it.
It's hormones. I was like, I remember doing it for something.
Yeah. So those are good? A gnome hat? And you want to make sure you've got them like, these antigens are like attached to the surface. They're not just like floating around. Sweet.
Kind of like that.
Mhm. Cool. All right, so everybody got those copied down? All right, so cool thing about the immune system, so you've got, how many different antigen shapes do you think there could be on viruses and bacteria? Not very many. In 3 dimensions? So it's probably like nearly infinite, because you've got, it's in 3 dimensions, first of all, and then size, and then, I mean, how many different 3D shapes can you make at a molecular level? Like almost infinite amount. So the challenge for your body is it's got to find a way to be able to recognize a nearly infinite number of shapes as foreign. So then what do? They make an infinite number of antigen binding receptor shapes. So that randomly, hopefully you have a shape that you made that fits one of those antigens. And you don't have to have one that fits every antigen, just, you know, at least one of the antigens that that virus or bacteria has, you recognize. And if it's a virus, it's got a decent amount. Bacteria have an enormous amount of antigens on their surface. So Viruses are a little harder. Maybe they've only got 3, so hopefully you can recognize one of them, or else you're in trouble. So you'll see how that works. First, little background. Your B cells, they're made in the bone marrow. And then once they mature and they're ready to function, they go to your lymph nodes, which are all over the body basically, and then the spleen. Your T cells, pretty similar. They're made in the bone marrow, and they go to the thymus to develop. And then when they're finished developing, they go to the lymph nodes and the spleen. And that's mostly completed by the time you're like 15 or so. So you could take your thymus out now and nothing bad would happen to you, because you're pretty much done making B cells. They're all in your lymph nodes and spleen already. All right, so when we say B cell development, one of the first things that has to happen is you make a whole bunch of B cells with different antigen binding receptors. So it's kind of a random process, the shapes they end up with. And you could see their little antigen binding receptors on the surface. And you could imagine, they could have all those shapes you made, those binding receptors. But you could imagine if you're making random shapes, do you think you could make a shape that would bind to your cells? If you're just making random shapes? You could, right? That would be bad. Now you've got autoimmunity, your body's killing its own cells. So before they're done developing, before you send them out into the body, to the lymph nodes, they're checked by special cells that kind of inspect these receptors, and they look for any that bind to your cells. So if they do bind to your cells, then they're told to kill themselves. So go into apoptosis, sorry, you developed not the right way, you don't get to live anymore. That's what happened. So they're eliminated. That's what these little X's are. Very important, because if you made B cells that attacked your own cells, you could die, basically. Or have your kidneys stop working, or who knows. So that's clonal deletion. After you do that, then those B cells go to the lymph nodes and wait for an infection to happen. All right, so which of these is true? The B cells that remain after that process. Anybody else? There's 14 of you in here. All right, looks like you have some discussing to do. These are good questions. All right, final votes. Maybe someone convinced you. Anybody else? 11. All right. So we're trying between C and E, and the answer is C. It's the only answer. And I put D on there because people know that B cells produce antibodies, but not right away. You'll see how that happens. It takes like 5 steps to get them to making antibodies. And then the reason they don't make antibodies right away is that would be a real waste of resources. So if you've got 100 million B cells all making antibodies, that's kind of pointless because most of them, those antibodies don't recognize anything. You only want to make antibodies against something that's trying to get you. Otherwise, you're just clogging up your blood with antibodies. All right. So what about the T cells? It's pretty similar. So the only difference is instead of developing in the bone marrow, they develop in the thymus. Does this slide look familiar? That's because it's the same process as the B cells went through. That's why we have the same picture. All right, so which of these is true of the T cells after clonal deletion? Did I put enough in? Yeah, I did 6, right? All right. So the correct answer is C again. And what did some other people put? E. So they don't recognize human cells at all. The ones that recognize human cells were killed in the deletion process. So only C there. All right, so in order to activate our B and T cells, we've got to get the antigen to the B and T cell. And the cell that does that is the antigen presenting cell. Most of the time, these are dendritic cells and macrophages. And if you notice, those are they in the first, second, or third line of defense? They're second. So you've got something infecting you. Those macrophages are trying to take care of it. They're eating those pathogens. Remember phagocytosis? But what if that's not good enough? What if you're not going to get rid of it just from macrophages? So they're able to activate the third line of defense. And the way they do it is they take antigens from that bacteria you drew and put them on their surface. So they can go show it to the B and T cells. Got a little movie of that, first 2 minutes. So that's a good volume.
Systems and accumulate inside the lymph node, spleen, and other secondary lymphoid tissues. Other types of cells are also present, including B cells and macrophages, both of which can also act as antigens and cells. Consider a macrophage. To counter an antigen which has no specific receptor, it can still engulf it using, for example, selected microreceptors. This is termed non-specific internalization. The pathogen bearing the antigen is contained within a phagolysosome in the macrophage.
So there's that lonely little virus that just got eaten.
Use the phagocyte, bringing proteolytic enzymes in.
So this is like the steps of phagocytosis that you saw in part 1. So break it on down.
Elsewhere in the macrophage, major histocompatibility complex molecules are being synthesized at the endoplasmic reticulum. MHC class II molecules are transported by the vesicles, which intersect the pathways of vesicles released from the phagolysosome. Proteolytic fragments of the antigen bind to the MHC class II molecules in a special compartment, displacing the invariant chain of the class II molecules.
You don't need to know all these details, by the way.
Loading is facilitated by another MHC-encoded molecule called EN. The vesicle containing the MHC class II antigen fragment combination migrates to the surface of the macrophage, where the contents remain attached, presented to any Th1 cells that make the contact.
So eat the virus, attach it to your little presenting receptor, and then put the antigen on top of it. And you can use that to go activate the specific immune system. All right, so how about with your group questions 3 and 4 on there? And remember, it's an APC ate your bacteria that you made with the Play-Doh out there and put one of the antigens on the surface. You're right. That's exactly right. Not inside it, after it presents it.
Oh, like after it binds and the thing comes out?
Uh-huh. So it'd eat this whole thing, nom nom nom.
Yeah, yum yum yum, back man.
And then it picks one of those to present on the outside.
And so do you, since, like the way it was in that video, is not like how we drew them?
Pick one of your antigens to present.
Yeah, I know, how do you draw that out there though? Like, because it doesn't look like that, like, because like when I'm thinking about like in your past slides, like this one for example, this is the little piece that it's presenting, right? Because
No,
it's
those
next to
are
it.
antigen binding receptors.
Oh, and that's it.
That's later.
So it like eats it.
Uh-huh. And then it takes this one and puts it on a little stick on the outside.
The outside of it.
Mm-hmm. That's it.
Oh my god. Okay.
And that's 3, right?
That's 3. For 3 and 4? Well, it's funny because you actually did 4 too.
Oh, really?
So 3 is just the triangle
Yeah, and then
sticking out, and then 4 is the thing on the outside.
I'm gonna draw it that way, just so I know.
Yeah, and remind you might want to write down on all these, like what's the antigen and what's the antigen binding receptor, just to label it, so when you look back at it, you know.
So is it this direction or is it that same direction we did?
So it's the blue thing. That's the antigen.
Right. But I was saying, so is it drawn this way or is it drawn exactly how we have it?
It'd be exactly who you have it.
So it's the antigen on the outside and the binding receptor on top.
Right,
Okay.
like that?
But we're doing it in 2 steps.
right.
So like that.
And if you want, you could put like a little stick on the end of it or something. Just remember there was that molecule that was holding it. So like,
Like putting that up.
which one are you doing, the triangle or?
Oh, I picked a triangle.
Okay. So like, there's your APC, your macrophage, and it put the antigen on the surface. And that's a little thing to hold it there. That it produce. So then see if you can do 4 or 2, which I think you did already.
The antigen binding receptor, is that part of, what is that attached to?
Eventually it'll be attached to B and T cells.
So it's not on the surface of B, like this is just there?
For now, we just wanted
Attached
the,
to it? Or
that's attached to a B cell.
so we're assuming that there's a B cell here.
Right.
Okay.
But for now, we just wanted to get the shape of it.
Right?
Make sure you label for this whole front part of the worksheet your antigens and antigen binding receptors.
Did I do 4 on this one? So this one would basically just be the.
It would be on the other side of it. So like, you took the square. So that one's right.
Over here.
No, so then there'd be this little green thing that you drew. That would come bind to it on the APC.
So like that, and then you have the one that binds.
Yeah, the green thing. Yeah. Right, exactly. See what I mean?
Yeah, okay.
So that sort of looks like it would fit, but
That wasn't very accurate.
so make sure for that 3 and 4 you label the antigen and then the antigen binding receptor, so you remember which one's which. So the orange is antigen, the green's antigen binding. All right.
This is our antigen, right?
Yes.
And then this is, okay, because I keep thinking of blood, and it's really screwing me up here.
Yeah, it's annoying because
Do you know what I mean?
you really
Like,
really
two
shouldn't
separate
call
complete-
them.
Yeah.
It's not separate, but we shouldn't call them antigens
Yeah.
on the blood.
And this is where
Probably.
I immediately go back to like the blood and our coagulation and stuff. And it's just screwing me
Yeah.
up. Okay. So this
Because
is
when
our-
it's part of your own blood cells, it's not really an antigen.
Right, right.
But if it's somebody else's
Okay,
blood,
yeah.
then it is an
Okay,
antigen.
so this little guy right here is an antigen.
Right. So the orange ones are antigens,
And
the
this-
green ones are antigen binding receptors.
Just so I can label this.
So you want to label those.
Antigen binding.
Uh-huh. And so attached to that thing you drew is a big circle.
This?
Yeah, like the bottom of it. And that's your macrophage, holding that out there. Right, and that's your macrophage. And then that circle thing, that's the antigen.
So do you want us to label those, not those?
Oh, I would label it everywhere just to remind
Okay,
yourself.
wait a second. I thought we just drew a stick off of our antigen.
You
She
drew a
also put the
stick?
binding receptor on it too.
Well, 'cause it's
No,
like-
that's the presenting part.
Yeah.
That's the stick. So that big circle, just like Annabelle has, that's the macrophage. Then it's got a stick that it presents the antigen with. And then there's something that binds the other side of it, like your green thing you drew. That's the antigen binding receptor. So you're almost there. Right? So then that black square is, yeah, that's the antigen. And then that white thing around it is the antigen binding receptor.
So to present it, like throws it back up?
Mm-hmm, exactly.
See, I was thinking it
Yeah,
just
it's like,
threw up,
blah.
I thought it just threw up the antigen.
It does.
And then another
Another cell's going to have that binding
receptor
receptor.
on it.
Right.
So why, I know this is probably stupid, but, so why does it have the receptor in the first place so that it can be spit out without it? To have it bind with another one? Does that make
You'll
sense?
see.
So that's what it's doing though.
Right, it's because
But
it
there's
wants.
another reason why.
Yeah, it
So like
wants
it
to go.
binds twice with the same thing, but in a different aspect. Because it binds right here. You have your binding receptor, but then it gets yum yum yum, and then it gets
This
spit back out
isn't.
with another receptor.
This isn't part of the macrophage.
But it's a whole different separate thing.
It's going to be the next step.
Okay.
So the macrophage just knows this isn't your cell. It just sees it with the orange stuff.
Okay.
You said throws back up only the antigen?
Right.
And then the macrophage?
And then it's going to go find something that has that antigen binding receptor a little bit. All right. So we've got our macrophages, our APCs. And so next up is going to activate the specific immune system. So this prize first. We'll get see you. So that antigen presenting cell, it's going to go find a T helper cell that has that antigen binding receptor that you drew, and activate that T helper cell. So what does a activated T helper cell do? It divides and produces more T helper cells that has the same receptor. Simple enough. So you're going to draw that for me, which is number 5. So what we're trying to do is turn all those words into a picture. So read through that one, chat with your group, see if you can get it all figured out. So make sure you read those instructions twice, I would say.
Well, a specific one would be 1, 2, 3, the 4th bubble.
Well, because it's got a, it's a specific.
Close to these two, so they'd be the ones divided.
I'd pick one of them, because I think really it fits better into one of them.
Because I don't think you can find any of the other ones. You don't think you can find this one?
Yeah, I would say the third one's the most specific shape.
Oh, drawn in tables. What happens if these cells were exposed to this antigen present?
Yeah.
And if you're not sure what's supposed to happen, just glance back at those. That's right. Uh-huh. So make sure you draw everything that happens on that slide.
So you would draw more of these.
Which one?
This one?
Yeah. Uh-huh. Yeah.
No, it's going off of this. Which one of those will this bind?
A few. Well, what does it say happens? Well,
Keep going.
well, what does divide mean?
Split her apart like.
Make more, right? Uh-huh. So draw a bunch of them.
So it would make one eye.
No, because it just says make more T helper cells. Oh, settle down.
Divide and produce more, so then.
Go ahead and make sure for all those you label like APCs and T helper cells. So when you go back, you know what those circles are.
So is it?
You're doing something slightly different. So you're showing me which APCs activate them, which is right. But what I was saying is, what if that APC comes?
This one. This is our APC. So which one would we use? We would. No, which one would we use? Do that one.
Mhm.
Yeah.
Right. Yeah. And I know what you're doing, so I would just cross it out, but because you're doing like which APC activates each of those, which makes sense, but we were talking about just that one APC up there.
Oh, I didn't know it was that.
We'll just do with that one.
That's right.
I was just reading it.
Yeah, yeah.
I'm like, okay.
Mhm.
But I didn't read the instructions.
That's okay.
Nothing would happen with
I don't
the
read
other ones.
instructions usually either.
Nothing would happen with these other ones.
Nothing would happen with this other one. And what would happen to that third one?
Would be activated.
And what happens when it's activated?
It makes more.
Mm-hmm.
Yeah, I made more. I didn't draw my one here though. Do you draw the same as it just says so?
Just making some babies.
I need to figure out how to remember that these cells are your.
All right, so you're good. All I would add is you want to label your APCs and T helper cells and antigens and antigen binding receptors on your picture. See, all are good. Are you peeking over
I
there?
was making sure I did it right. Okay.
That's as good as my drawing gets.
They're not connected together
I
though.
like this.
It's like that.
So is this one?
And then once you got those pictures, I want to see APC, T helper, antigen, antigen binding receptor all labeled. Cool. You gonna draw some more of those?
More of these?
No. What makes? What do you get more of? More APCs or more T helper cells?
Then this is
So which one are you gonna draw more of?
binding receptor, right? This one?
Yeah.
The bottom part, this is the antigen binding receptor. right?
Uh-huh.
This is an APC and this is a T helper.
Mhm. We'll see, right? So if you just draw like 3 more of those, then you'll be good. 3 more T helper cells. What? I want extra credit for that.
But you can get up to 30 points.
Who lets you get 30 points extra credit? All right, so yeah, what's that thing sticking out? Antigen or antigen binding receptor? Yeah. So like those little.
So is this one the antigen receptor or is it this?
The second thing you pointed at. Uh-huh. That triangle is the antigen. Like that. You want to label that thing the antigen. And then the cell it's attached to, that's the antigen presenting cell.
But then you tell certain people it's all about.
All right. All right. So everybody got those? And what those T helper cells do now is activate B cells that match the antigen, which we'll see in a little bit. They also release cytokines that turn on inflammation, and cytokines that activate macrophages, and things like that. And don't worry about the CD4 thing. That has to do with HIV. Because HIV targets those T helper cells because they have that receptor. All right, so we'll pick up with this tomorrow during lab time. Should be fun. Oh, and be a good child and put your Play-Doh away so it doesn't dry out.